So the test is going to be on Thursday, as you know. Okay, initially I posed the wrong day. I said Wednesday, but that's for my other class. For you guys, it's Thursday. I just found out in the morning you guys also have a math test, but that's okay. That's okay. You get the break to look forward to immediately after this. So you know, just work hard and you'll be fine. And the test is really straightforward. Okay, the study guide, this test review should be your main focus. Okay. Everything in this guide is going to show up on the test one form or the other. Okay. So you must know everything when you are preparing. So I just returned the lab to you. You just have to find your group members. Some of you, you know, if you have a really less than ideal grade, I gave you the opportunity to send me back and fix some of the questions. You can do that by the end of the week, and it's okay. Right? Focus on the test first, and then you know do the lab adjustment later on. Okay, so speaking of the Beetroot lab, here are a few questions that you have to know. These are the only questions you have to know in preparation for your tests on Thursday. So first question. What is the control of the experiment? Control of the experiment is the test tube with the room temperature water, right? Okay. Room temperature water. The purpose of control is to provide a reference point to compare your treated groups with, right? To provide a reference, provide a baseline to compare your test results with. You can pick up a hard copy of this review sheet at the front, okay? Right at the door. All right, next question. What is the dependent and the independent variable? Okay, so for dependent variable, that would be the color intensity, right? Color intensity. Not the absorbance of green light, because we actually didn't do it, right? The absorbance was just data that I gave you, right? In the actual experiment, we look at the color and compare it visually. Right? That is an example of qualitative analysis, right? Whereas the independent variable, that's going to be temperature. Okay. What variables are standardized? There are many things that were standardized. For example, like the size of beetroot, right? Time left in the water, time that the beetroot, right? We, for example, only left the beetroot in the water for how long was it? Like 1 minute, right? I think. Yeah. And then when you put it in the test tube, it was for like 10 minutes or 20 minutes, right? Okay, so that's standardized. Of course, if you leave it in for longer, more color is going to seep out, right? So we want that to be the same. Size and shape of test tube, right? Water level in test tube. Okay. So there are a bunch. Why does a frozen beetroot have similar color intensity as 80 degrees when placed at room temperature? I told you in class, freezing destroys the cell membrane? Pigments will leak out. Will leak out even at room temperature, right? You don't have to heat it up. It's already damaged. Normally, you heat it up to damage the cell membrane, right? But now the freezing actually also damages the cell membrane. And when you put it at room temperature, all the color will just leak down. And it's going to be a very intense color, similar to that of the 80-degree sample. Pigments will leak out even at room temperature. You just stop me if you have questions, okay, if I'm going too fast or something like that. So the graph was probably the most problematic question for a lot of the groups. There's a video that shows you how to do it, okay? So, watch it. But obviously, you're not going to have to graph on the test. This question does ask, what do you expect the absorbance to be if we included temperatures higher than 80 degrees? So your graph looks something like this, right? Looks something like this, right? And this is about 80 degrees, right? About 80 degrees. So what the question is asking, like what happens if you heat it up to like 90 degrees, 100 degrees? What do you think will happen to the absorbance? Do you expect it to continue to go up? Do you expect it to be about the same? Do you expect it to come down? What do you think? Is it 1, 2, or 3? What do you think? Oh, sure. I'll post it. Don't worry. Yeah, I'll post it. So if you don't have to take pictures of it. I'll post this afterwards. What do you think? If we go to a higher temperature, 90, 100, do you expect the absorbance to continue to rise, to be kind of flat, or it's going to start coming down? Huh? 1? Yeah, it's gonna be flat, guys. Okay. If you look at 60 to 80, it's already very similar, right? If you remember your experiment, the greatest change in color was between, I would say, around 60 to 70, but 70 and 80 are very similar, right? Okay. Because at that point, at 70 and 80, the membrane is already destroyed, right? If you heat it up more, it's not going to destroy it more. It's already completely destroyed by that point, right? So that's why going higher temperature will not change the absorbance, okay? So like this, right? No significant change. Because the membrane is already completely destroyed. You cannot get more pigment out of it. Everything that needed to leak out had already leaked out. Do you understand that? Now, here's the moment you guys all been waiting for. There are only 3 multiple choice questions on the lab. If you understood all of these things that I just told you, you'll be fine. Okay? Just 3 multiple choice questions for the lab. All right, moving on. We just have a lot to go through, so sorry I'm rushing a little bit, but like I said, everything will be posted online afterwards. List of characteristics of living things. I mean, this is one of the first thing we learned, right? So like organization, you know, ability to maintain homeostasis, growth and development, reproduction, energy requirement, respond to stimulus, and there's one more. What's the last one? Yeah, that's right. The seahorse, right? Remember? Adapt and evolve. So, I could ask you which of the following is an example of characteristics of living things, and you have to recognize them. Okay. I can also ask you like the other way around, which of the following is not a characteristics of living things, right? Next, what is a placebo? Placebo is a sham treatment. It's a sham treatment with no effects on the dependent variable. Do we always need a placebo when we do experiment? No, only when we deal with who? Us, right? Only when we deal with humans, right? Okay. So, give an example where placebo is needed. Okay. So involving humans. E.g. testing new drugs, right? Not needed would be like testing like driving with window open or closed, right? Versus fuel consumption. Right? Sorry. Which one can you not read? The green one? The red. Testing new drugs. Sorry. Okay, hold on. Let me change the green. Okay. It looks better on my screen. Okay, so driving with windows open or closed versus fuel consumption, right? You don't need that you don't need a placebo for that, right? There's no bias to minimize. There's no psychological effect to counteract. Everything is right as rain. Okay, what is a valid hypothesis? That's like a question from the first quiz, right? You should also go through your quizzes. Right? Don't do this psychological thing with me. Don't be like, oh, he put this question on the quiz, he won't do it again. I will. Okay. So go through your multiple choice, go through those short answer questions. Valid hypothesis is testable, right? Yeah. And you should also know a hypothesis is a tentative answer, right? to question. So just remember, the hypothesis shouldn't be another question. Okay, it's a statement, right? Increasing the temperature will increase the membrane permeability. That's a hypothesis. Okay, you don't have to do explanation. You don't have to give reason. You just state what you think is going to happen. Okay. Let's take a look at the graph. I have not tested you on the graphs yet, right? Remember? So there will be a question related to this. Let's take a look at the one on the left side. Okay, we have the placebo group, also known as the control group in this context, and we have the treatment group. Does the drug work or not work over here on the left side? Work or not work? 1 for work, 2 for not work. Okay, show me what you think. 1 or 2, work or not work? Okay, good, good, good. Okay, yeah. Anyone else over here? Okay, good. Everybody said it doesn't work, and that's correct. Okay, so this is ineffective. Ineffective. Why? Because they both work equally well. It's not the drug that's working, right? It's just the fact that you're taking something that makes you feel better. Okay, so both of these, both groups have the same effect. Right? It doesn't matter that you are taking the drug or not. That means the drug is not working, right? This one is effective, right? Only treatment group has a reduction in symptoms. Okay. And that is it for lecture 1. Okay. That's all the things you have to study. Okay. The purple stuff will not be on. And so no experiment with the flies and the swan neck and all those things. Okay. Any questions about lecture 1? Okay, 1 down, 4 to go. Lecture 2. You probably want to copy this on a separate piece of paper just because there's like, too many things. All right, let's see if this is going to work. For carbohydrates, what are the elements? Just yell them out. Yeah, I think I heard these 3 things. Carbon, hydrogen, oxygen. As for monomers, we have monosaccharide, which we only did 3 types. Right? There are the glucose, galactose, and what's the last one? Fructose. And of course, they could join together to form double sugar, right? Disaccharide. Biological function, we had 3. We did 3 things, 3 or 4. There is the glycogen. That's energy storage in animals. I'll just do the example and the function together, okay? What's the one that is storage for plants? Starch. Yeah. And then there is cellulose, which is, yeah, the cell wall, right? And then the last one is chitin. Chitin. Shells and exoskeleton. Bless you. So yeah, I'm just joining the last two columns together, okay? All right. Proteins, what do we have? Yeah, CHO, and then what else? And traces of sulfur, right? A little bit of sulfur. The monomer is amino acid. Amino acid. I'm not going to ask you to draw amino acid. 20, right? Biological function. So they could be for energy source. Okay. If you run out of carbohydrates, you can burn some proteins. What else do we need proteins for? Anybody? What's that? For okay, hormones, that's right. Hormones. Such as insulin, right? What's another function for protein? Enzymes, yeah. Which is to catalyze chemical reactions, right? To speed up chemical reactions. And example of that would be like lactase, which breaks down lactose. What kind of things in the body are made up of proteins? Yeah, muscle, nails, hair, yeah. So these are the main function that we learned for proteins. The test is not difficult. It's just a lot of information to remember. But the questions themselves are not tricky, I hope. No, they're not. They're not. I mean, I hope you would agree. Yeah, that's what I meant. It's straight out of this package, okay? All right, next we have the lipids, which is CHO, and then some trace of P, right? Traces of P. But not all of them, right? Only the phospholipid has the phosphorus, right? Okay, so the components include things like glycerol, fatty acids, phosphate group, carbon rings, right? These are the key things that we could use to build different kinds of lipids. We use lipids for energy storage. This is the triglyceride. We can use it for cell membrane structure. There are two things that are found in the cell membrane, which are lipids, right? Do you guys know what they are? There's the phospholipid, and what's the other lipid that we find in the cell membrane? Two more functions for the lipids when you're ready. What else did I do? What's that? Insulation, yeah. Sure, that's not what I was thinking, but that's totally correct. You see, I did not study for my own test. Another one, maybe? Wax, yeah, it forms water resistant barrier. Okay, there is one I really want to have. Hormones, that's right. I don't know if I gave you any. Oh, I did, yeah. Like testosterone, right? Like estrogen. Okay. Now I want to point this out to you in case you didn't clue in. Hormones can be proteins or lipids, right? Steroids specifically. Okay. These steroid hormones are made by the smooth ER. The protein hormones are made by rough ER with the ribosomes, of course. Okay, I think that's good enough for now. Let's move on. Nucleic acid, CHO, N and P, right? Nucleotide. Just to refresh your memory, a nucleotide contains phosphate, sugar, plus nitrogen base. All right, so for DNA, it's ACTG. And then ACUG. The function DNA contains instruction to make proteins, right? RNA, it makes up part of the ribosome. Right? And it plays a role in transcription. That's right. Okay, that's all we have to know for now. In terms of the function for RNA. We'll learn more about it in the second half of the course. Okay, what's this first one? You should be able to tell me. We have the phosphate, the sugar, and the nitrogen base. Nucleotide, that's right. There's a sheet at the front, James. This one here. Yeah, some kind of sugar, right? Okay, not sure which one it is. I mean, like it's glucose, but then like you don't have to know. You can just say sugar. And then this one with the rings. And again, right, you should download all the poll questions that we did in class. The answers at the bottom of the PowerPoint. A good 5 or 6 of those questions will be on the test. Right, so make sure you go through them yourself. Do you guys know what I'm talking about? No? Okay. If you don't know, that's okay. Let me show you quickly. So here it says wait, did I not show you? Oh, maybe I didn't. It's there somewhere. Yeah. Sorry. Okay, so you just go to content. Maybe I didn't do announcement. Content. Sorry. Test review. And then here. Test 1 poll question collection. Okay. That's like all the poll questions we did in class. Okay. It's like 50 something, 46 of them, okay? You cannot see the answer here, but then after you download it, it's gonna be like at the bottom of the PowerPoint. Let me see if I can download it. You see, once you download it, the answer is gonna be at the bottom, okay? So make sure you go through them. I mean, it's not the first thing I would be studying from, but I would definitely go through it. Yeah. I think 5 or 6 questions is from there directly. Okay. Compare similarities and differences between saturated and unsaturated fatty acids. Okay, so let's do this. So, in terms of like the structure, right? So, this one is going to be actually, let's just do fat instead of fatty acid, okay? Fats. Fatty acid is just the tail, right? Fat is like the whole thing, right? Okay, so for Let's say we compare composition. For saturated fat, it is 1 glycerol plus 3 saturated fatty acid. Okay, I'm just going to switch color for the other one so they don't look too similar. This one is 1 glycerol plus at least 1 unsaturated, right? At least 1 unsaturated fatty acid. Okay, let's take a look at the carbon and hydrogen. For saturated fat, carbon atoms are connected to maximum number of hydrogen, right? Whereas the unsaturated fatty acid is not. Okay? If you memorize one, you will remember the other one. It's just completely opposite, right? Let's do fatty acid structure. So this one is like, straight, right? Like zigzag, but straight. Okay. So no double bond. No kink. No kinks. There's no sharp turn. That's what kinks means, right? So this one has double bond. And kinks. Okay. There is one more thing we should compare between the two. What is it? That's right. So is it solid or is it liquid, right? At room temperature? State at room temperature. For saturated, what is it? Is it solid or liquid? Solid. Example, butter, huh? Lard. This one is liquid. And just name your favorite oil. Sure. Yeah, fish oil, sure. The one with the orange flavor was good. Are we good with the comparison? I would know this if I were you, okay? I would know the whole thing, but then if you have limited space, right? This one, I must know. Okay. Next question. If you supply radioactive nitrogen atom, if you don't know what that means, that's okay, because in bracket it says it's something that you can trace and detect. What molecules will you expect to find the radioactive labels in? This is just a very indirect way of asking you what molecules contains nitrogen. Okay, so of the 4 macromolecules that we talked about, which one has nitrogen? Yeah. Proteins and nucleic acid. These are the molecules that have nitrogen. You're not gonna find it in, you know, glycogen or something. Glycogen only has CH and O, right? Okay, moving on. We already talked about the monomer of DNA, which is the nucleotide, right? So I'm not gonna go through that again. How are the nitrogen base classified? Well, we know there are the purines. Purines have 2 rings, right? And they are A and G. I keep on telling you to remember pure as gold. And purine has 2 syllables, so 2 rings. And the other one is pyrimidine. How many rings? Yeah. Cytosine. It's okay to write short form for ACTG on the test. You don't have to spell it out. Like on the quiz, I give you like this little space. Many of you try to pack the whole name in, which was okay, but like, you know, you didn't need to. Base pairing rule. What does A pair with? A pairs with T, right? With 2 bonds. C pairs with G with 3 bonds. I will ask you to calculate again. So something like this, 30% A, okay, and you have to figure out the rest. So 30% A, how many percent T? Same, right? They add up to 60, which means you have 40% left. Divided by 2, 20C and 20G. Okay. See, uracil? There's no uracil in DNA, remember? Okay, what force holds the nitrogenous base together? The hydrogen bond, right? What is the significance of base pairing rule? Yeah, DNA strands are complementary. If you know one strand, you can predict the other strand. This is going to be very important in the second half of the course. Okay, random question not related to DNA now. Okay, this is a completely different question. Denaturation versus dehydration synthesis. Denaturation is what happens when you break down a protein structure, right? Using heat, changing pH, changing the solution. Example would be like cooking an egg. Right? The egg white goes from transparent to turning white. That's denaturation. Okay. Dehydration synthesis is like when you have two monomers and you connect them together, right? They are completely different concepts. Okay. So no questions on the 4 levels, the primary, secondary, tertiary, you don't have to know that. You don't have to know how an enzyme works, but you should like know enzyme is a protein, okay? I'm not gonna ask you to compare DNA and RNA. I already asked you that on the quiz. You don't have to draw anything for this unit. Okay, for this lecture. Any questions about number 2? Okay, for the interest of time, I'm not gonna go through this one. Okay, you already have it in the lecture guide, study guide, right? You have to know the functions of all the organelles listed here. Okay, I'll tell you what the question is. Okay, you don't have to label, but I'm gonna give you a description of something. Let's say this is this produces energy, and you have to tell me which organelle is it. That's right. If you didn't know that, good luck. But it's mitochondria. And I will give you a word bank to choose from as well. Okay. So nice, right? Okay. Previously, people didn't see the word bank. Okay. But this time I use font 25, so you can't miss it. Okay. Take advantage of the word bank. But these are the only ones you have to know. You don't have to know centrioles. You don't have to know peroxisome. Okay. You don't have to know any of those cytoskeleton, which I know you don't like. And then, yeah, there's no labeling either. Okay, done. Moving on. Circle higher or lower for each factor that will correspond to increasing and decreasing membrane fluidity. If you want to increase membrane fluidity, what are we gonna do? Do we want more cholesterol or less cholesterol? Oh, that's okay. More, okay, more cholesterol, more cholesterol. You stick more cholesterol into the membrane, the phospholipids are forced to be further apart, right? And so it's more liquid that way. Based on your lab, do you want higher temperature or lower temperature if you want to increase fluidity? Higher temperature. More unsaturated fat, because unsaturated fat is the one that's liquid, right? And then less saturated fat. Okay, so high, 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 low, and then low, 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 high for the other. Okay, list functions of membrane protein. Let me just get my pen back here. So attachment. To cytoskeleton. Enzymatic activity. Cell recognition. Cell signaling. Cell signaling. Transport. And something called intercellular junction. It just means staple two cells together, right? Please memorize at least 4 of them, okay? You don't have to explain, you just have to, remember what they are. All right, this one, right? Remember I did a really big chart with you in class? Let me see if I still have that note. Oh, there it is. Okay, so you already have the chart. I'm not going to redo it again. Okay, you have to review this yourself. You have to know which belongs to what category. So for example, a couple of things I can ask you, right? Which of the following is an example of passive transport? Then I can give you like exocytosis, protein pump, carrier protein, and you have to pick out from the list which one belongs to passive. And which one belongs to active. If I ask you which one involves a membrane protein, and it will be like channel protein, carrier protein, right? Protein pump, right? Not exocytosis, right? No, well, for endocytosis, the receptor one that requires membrane protein, but not the phagocytosis phenotype. I can also ask you, these are all possible questions, okay, so listen carefully. I can also ask you molecules like CO2, which is nonpolar and small, will likely cross the cell membrane using which method? CO2 will likely cross using what method? What is the chart value? Okay. So everything in this table. Right. You don't have to memorize the details, okay? Like, you don't have to remember the 3 and the 2. Should be straightforward. Okay, what are some factors that affect the rate of diffusion? Anybody remember anything at all? Yeah, size, temperature, charge, and the steepness. That's right. Steepness of concentration gradient. Like how much of a difference are we talking about? Is it 10% and 20%? That's not much of a difference. So you would expect it to be very slow. Talk about 10% versus 90%, that's quite a difference. And the diffusion is going to happen more rapidly. Okay, so this thing here, I already explained to you in class. I want to explain to you again. First of all, the membrane is selectively permeable. So the dots cannot move. Only the water can move. So let's get this straight. Right? Which way is the water going to move? Is it left or right? Show me what you think. Good. Anyone else? What was it, Genesis? That's it. Yeah, okay, good. That's right. It's gonna go to the right, right? It's gonna go to the right. So that's the first thing you have to remember. Okay, water moves to the right. So as time goes on, the water level goes up, right? On the right. And then the water level drops on the left. This gets diluted. And then this gets concentrated. It will continue to happen until when? That's right. It will continue to suck water to the right side until the concentration are the same. Until the concentration reach equilibrium. Okay, very good. Okay, this stuff, let's take a look. So first we have hypertonic here. Hypertonic. The solute concentration in the solution is blank the solute concentration in the cell. If it's hypertonic, is it higher or lower? Higher, that's right. It's greater than. That's what hyper means. Hyper means too much. So the rate of water entering the cell is less than rate of water leaving the cell because there's more solute on the outside, right? So let's take a look at an animal cell. This is before. This is after. I know I only give you one beaker in the package, but that's okay. You can draw an extra one. So this is before. Does that make sense? You can add more dots, okay, to emphasize. And so what happens is it will you will have more water leaving the cell. Okay, leaving. And then you will have some coming in. But the point is you have more water leaving than coming in, right? And then as a result, what happens to your cell? Does it get bigger or smaller? That's right. It shrinks. Okay. This is called plasmol and then lysis. Plasmolysis. Okay, for isotonic, the solution outside and inside are the same, right? Is equal to. And the rate of water moving in and out are also equal to each other. Right, so equal. In and out. Nothing too exciting. before and after looks the same. Looks the same. Okay, no change. No name either. Just not normal. And the last one is hypotonic, right? Hypotonic is when the solution outside is less than the concentration inside, but then the water coming in is greater than. So there's the cell. And then you have more solute on the inside than outside. So lots of water coming in and a little bit leaving. If this is an animal cell, what's going to happen? Burst. That's right. So lysis. Burst. Explode. Okay. There you go. No questions on catabolism, anabolism, and no question on IV either. Everything else you should know. Okay, lecture 5. I know there's a lot of stuff going on in lecture 5. So first, let me tell you what's not on. Well, this part I didn't even teach you, right? Like you had to watch the recording, right? It was like a sick day. So none of that. No questions on the poisons. No questions on the fermentation either. Okay. You will not be asked to explain the ETC or any of the cycles. Okay. I won't ask you to describe. I will not ask you to describe Krebs cycle or glycolysis. You just have to understand how it works to answer most of the questions. Okay. So what do you have to understand? Well, you should know how to write a balanced chemical equation for cellular respiration. Okay. This is probably the easiest thing that you can remember, but many people do not. Okay, so glucose plus 6O2. What does that produce? 6 carbon dioxide, 6 water, and don't forget the ATP. By now, you should also know which gets oxidized and which gets reduced. Does the glucose get oxidized or reduced? That's right. It gets oxidized into carbon dioxide. We have written this down 3 or 4 times now. Reduce. Okay, bring some colors to the test. It will probably help you, make the solution a little bit easier to read. I mean, you don't have to, okay? But you could. And calculator is also okay with me. Okay? Don't use your phone though, okay? If you're gonna use your calculator, use like a regular calculator. So why do you need your calculator? Well, you will need calculator for something like this. Assume a mitochondrion contains 50 NADH and 19 FADH2. If all the energy carriers were used to make ATP, what is the maximum number of ATP? You should know how to do this. Plug it in and tell me. 108? Oh, sorry. 188? Anyone else? You guys know how to do this? So 50 NADH, right? You should know by now. You multiply by 3, which is 150. 19 FADH2 times 2, right? You add them together. 180. Most of you probably don't need a calculator for this, okay? But if it makes you feel better, bring one. Good to have these psychological blankets. Okay, this was the question that was like I put many stars on during the lecture, right? Remember that slide? I said you must know this. So go ahead, work it out. Take it up with you in a moment. How many? 108? 108? Huh? Is it 108? No? You're not sure? How many? Not sure? Okay, if I show you how to do it, will you guys learn how to do it properly, please? Yes. What is it? Huh? One cycle what? One cycle is 12, yeah. That's right. But you cannot just put 12 multiplied by 9 on the test, okay? Some people are like, they try to outsmart me. They're like, okay, I can't memorize the whole thing. I'll just remember 12. And then I'll multiply by whatever number he gives me, okay? I will only give you like very little mark for that. You got to show me what's going on, okay? Hold on. So one cycle. Okay. How many NADH for one cycle? 3, yeah. And 1 FADH2 and 1 ATP, right? By substrate level phosphorylation. This translates to 9 ATP, right? Because each NADH can make 3. Times 2. So these are from oxidative phosphorylation. So that's how you get the 12 per cycle. If all the energy carrier produced in the prep cycle goes to ETC and becomes ATP, right, you will get 11 plus the extra one here, 12. You guys all understand where the 12 comes from? Yeah, but this is like, it says 9 turns, right? It turns twice per glucose. So you start with 1 glucose, then it will turn twice. Okay? 2 turns. 1 glucose, 2 turns, right? But this is not asking you what happens if you have 9 glucose, right? This is telling you if the Krebs cycle turns 9 times on its own.
1.
That's right. 1. Right. You got the 12? Yeah. Okay. Then we times the 9, which I think is 108. Okay. Again, I would know this along with, you know, the table that I told you about. Okay. So you can kind of guess, you know, what are the written questions, right? If you have been listening actively, you would have known a few of those questions already. If you haven't been listening actively, that's okay. I have the recording. You can go home and listen to it again. Okay. Which step of cellular respiration generate the greatest number of ATP? ETC, right? ETC. Describe what happens as H+ is pumped from matrix into the intermembrane space. Now, I know I said I'm not going to ask you to describe anything, but this question is important because of this. Hold on. Where is this? There it is. So that question was particularly important. I don't know if you guys watched the recordings. It doesn't look like all of you have though. Okay, I checked the number of views. It was in the low 15. So probably some of you have not had a chance to watch that, but that's okay. There's this question here in the lecture notes. I'll read it to you. Okay. Hydrogen cyanide is poisonous. It binds to one of the membrane proteins in the ETC and blocks the movement of electrons. If cyanide poisoning occurs, what would happen to the pH of the intermembrane space? Hint: low pH is caused by higher H+. All right. So let me just try to do this on the board. Like hopefully you know by now that we have the mitochondria. These are the ETC, right? And normally what we do is we pump H+ here, right? That's what happens when there's no poisoning, okay? And now they're saying in cyanide poisoning, you block the ETC, right? Okay. So when you block the ETC, do we pump the H+? No. Okay, so less H+. Okay. So what do you think? Less H+ means higher pH or lower pH? Higher pH. Yes. Okay. It says here, right? Low pH is more H+, right? Yeah. More H+ makes it acidic. Acidic is less. Okay. So because the cyanide blocks the ETC, we don't let the H+ go. We have less H+. That's why the pH is going to be high. Okay? You got to think this through. Okay? Don't just memorize high pH. Because on the test, I might not be asking you about the intermembrane space. I might be asking you about the matrix, right? Okay. What will happen to the pH in the matrix if you block the ETC? Well, if you block the ETC, the H+ did not go over, right? The H+ stays here. More H+ in the matrix means less pH. Okay, lower pH. Okay, think that through, please. Last one. What is the purpose of the final electron acceptor? That's like the parent who needs to pull the kid off the slide, remember? Oxygen is the final electron acceptor that will remove the electron from the ETC, right? And why do you need to do it? Allows continuous electron flow. Okay. That's all. Let me stop the recording here.